This is a production of Cornell University.
And so, as Bob said, what I'd like to do to talk to you today is recent work that we've been doing, essentially testing the hypothesis that plants have overinvested in chlorophyll. And so I will get into that both from the standpoint of theory and experiment. And it's an ongoing project, and so we don't necessarily have an answer, but I'll tell you about why we're interested in doing it. Before doing that, I guess I'd like to step back and just try to put this kind of research into a more global context, a context of really the challenges that agriculture is going to be facing over the next 3 to 4 decades. And the place I wanted to start is here. Many of you will recognize this as a textbook figure of the global carbon cycle. In fact, it's only half of the global carbon cycle. It has to do with the terrestrial global carbon cycle. And I picked that out particularly because, of course, that's where most of our food comes from, the terrestrial side. And what it shows is that this process of photosynthesis removes amount of carbon from the atmosphere that's equivalent to 15% of all the CO2 in the atmosphere. You know, and CO2 in the atmosphere is about 400 parts per million, and there's about 800 gigatons of carbon in the atmosphere. And so of that 120 gigatons that are taken out, about 60 are essentially very quickly returned to the atmosphere by autotrophic respiration of plants. And so this is the amount of photosynthate the plants use to grow and to maintain themselves. And what's left is approximately another 60 gigatons, and for all practical purposes, it supports everything else on the planet. And so it supports microbial life, and it supports all kinds of animal life. Of course, part of that is us feeding ourselves and feeding our livestock. And so the difference between this gross photosynthesis and this net photosynthesis is often called net primary productivity. And so the net primary productivity of the terrestrial biome is estimated, I have here at 60, but it's actually estimated at about 56 gigatons of carbon per year. And so I think it's informative to think about, what are those biomes, individual biomes across the world that support that net primary productivity. And you're probably not surprised to find out that the tropical rainforests account for about 1/3 of it. The vast savannas that are deployed around the world account for about 1/3 of it. But what may surprise you is that already the cultivation of the crops that we use to eat and to feed our livestock account for about 15% of the net primary productivity of the terrestrial biosphere. And so that's a pretty big carbon footprint. And so the UN FAO, the UN Food and Agriculture Organization, as well as several non-governmental agencies, are predicting that somewhere between 2050 and 2060, we'll need to increase global crop production by 70 to 120% in order to meet agricultural demand. Agricultural demand that's being driven by population increase, as well as many of the large population countries in the world becoming more affluent and eating further up the trophic scale. And so this seems like a huge challenge to as much as double crop production in the next 30 to 40 years. It's been done before. And so between 1960 and 2005, population on the planet increased 110%. So it went from about 3 billion to 6.5 billion. During that same 45 years, global crop production increased an incredible 165%, went from 1.8 billion tons to 4.8 billion tons. And of course, this was called the Green Revolution. One of the important things to know about that is 135% of that 165% increase was due to intensification. It was due to producing more per land area. And only 25% of that 165% was recruiting more land into agriculture. What this graph shows is that, at least for the land that's under grain production in the world, it peaked somewhere in the middle 1980s to the 1990s. And it's either been declining slightly or have plateaued over that time. And so apparently what that means is that if we're going to have to double crop production, then we're going to have to do it on approximately the same 120 million hectares that are under cultivation today. And so that means really doubling productivity on a land area basis. And that also means then that this carbon footprint for cultivation goes from 15% to about 25%, a quarter of all terrestrial carbon fixation to feed ourselves and to feed our livestock. Interestingly, too, if you figure in that we're going to do it on the same amount of land, that suggests that net primary productivity of the terrestrial biome has to increase by about 12%. And so, you know, I think this has to qualify as a grand challenge if we're going to meet this projected agricultural demand to actually increase net primary productivity of the planet through cultivation of crops. Unfortunately, it doesn't appear like things are going in the direction to be able to meet that demand as we're currently practicing agriculture. And so the way to read this figure is that for the 10-year period ending in 1977, so from 1967 to 1977, there was a decadal increase of about 30% per year in the increase of yield for these three crops, maize, rice, and wheat. And so these are the three major crops by production in the world. So about 3% per year. That was sustained through the 80s, but by the 90s, you see this dropping, and by 2007, which is the last complete data set, you can see for rice and wheat, big drops. So far, there hasn't been a drop in maize. And I think another interesting thing to think about is that China is the biggest rice producer in the world, and also the biggest rice consumer. Right now, they're investing 1,000 times more in rice research than they did in the 70s and the 80s. And so it suggests us to be able to sustain any kind of increase that low-hanging fruit might be gone. And that we need to look for different kinds of traits to optimize in order to create the situation where we could really think about increasing crop productivity on the order of a factor of 2. And so what is the cause of that stagnation? One of the causes is almost certainly a stagnation in yield potential. And so yield potential is the genetic potential of a crop. And so it's defined as planting a crop in a situation where that crop's well adapted and is not limited by the availability of water, the availability of nutrients. It's not beset by pests and pathogens, and it's free from lodging. And so it's really for a given growing area that the crop's adapted, it's the highest yield that you can possibly get. Yield potential is defined, first of all, and is constrained by the amount of solar radiation that's available during the growing season. And so the amount of solar radiation that's there to drive photosynthesis is the total amount of energy that's available. And then there are 3 genetically determined things in the crop that act on that then yield what the yield potential of the crop is. The first is the efficiency of the crop in intercepting this available radiation. The second is the efficiency of photosynthesis in converting this intercepted radiation into biomass. And then the third is the decision that the crop makes in how much of that above ground biomass it's partitioning into the part of the plant that we care about. And so the wheat grain or the maize grain or the seed grain. And so what happened in the Green Revolution in that 165% increase that happened in the period that I talked about was really driven by 2 factors here in terms of yield potential. The first is interception efficiency. And so breeders were very good at selecting for crops that covered the ground very, very quickly. And so get that first leaf layer out, because any radiation that hits the ground certainly isn't going to grow a crop. And the second is that really drove it in the small grains is this partition efficiency or the harvest index. And so now our best crops for soybean and rice and wheat, 60% of all above ground dry matter is already partitioned into the grain itself. And so this value now is, once a canopy is formed and across the entire growing season, our best crops are intercepting about 80% of the total radiation that's available. We think the theoretical upper limit might be 85 or 90%, but clearly there isn't the capacity to really significantly increase the overall yield potential of our best crops. This being at 60%, we think that the upper limit of that is probably between 65 and 70%. You need pods, you need leaves, and so not everything can go into the seed. What didn't contribute at all to the green revolution was any improvement in this conversion efficiency, the efficiency of photosynthesis. And so the question becomes, is there room in terms of increasing the efficiency of photosynthesis that would bring us into this realm of being able to significantly increase yield potential? And so the way to get at that, we think, is probably to try to estimate what the upper efficiency of photosynthesis is. And so we weren't the first to do this calculation, but let's suppose that we start out with 1,000 millijoules of sunlight. And so that's all of sunlight, the entire solar spectrum. Photosynthesis is driven by essentially the part of the spectrum that we can see. So from 400 nanometers to 700 nanometers, which means about 50% of the quanta that are available in total sunlight are outside of the photosynthetically active spectrum. So immediately you have a 50% reduction in the potential efficiency. Plants are green. So they're green because green light isn't absorbed very well, and we'll talk a bit more about this later, and is reflected to our eyes, and so there's loss there. Early in the season, some of it's transmitted down to the ground, and so there's a loss there. This photochemical efficiency has to do with the fact that even though a blue photon has 1.75 times more energy than a red photon, all photosynthesis is driven by the energy of a red photon, and so that extra energy in the blue band is completely lost to the heat in femtoseconds, and so isn't available for photosynthesis. Carbohydrate biosynthesis is exergonic. There's this process of photorespiration in C3 plants. And then finally, there is autotrophic respiration, this respiration that we talked about, plants growing themselves. And so when you get through to this, it says that the upper potential for photosynthetic efficiency in a C3 plant, like a soybean plant or a wheat plant, is about 5%. C4 plants are a bit higher. So plants like maize and sugarcane can do about 6.5 to 7%. The maximum that's ever been observed across an entire growing season is about half of that. And so this comes from these farmer competition trials where, where they grow these small plots in competition for the highest yield. And the average that's observed for, say, high input farmers in the Midwest is about 1%. And so it suggests this difference between 2.5 and 5, or this 1.5 and 2.5, is an opportunity to significantly increase yield potential. And so why is photosynthesis so inefficient? So why is it operating at 1% or 2.5% rather than the theoretical 5%? There's a number of reasons, but the one I want to talk to about today is this one. And that is, this is the dependence of photosynthesis of carbon gain in the plant on the intensity of light that's available. And so this is in micromoles per meter squared per second. And so, in Champaign-Urbana during the growing season, the maximum light intensity is about 2,000 microeinsteins per meter. Again, in Ithaca, it's probably down here somewhere. But what this shows is that photosynthesis in a soybean plant or a wheat plant saturates at about 20 to 25% of full sunlight. On a sunny day, at the top of a soybean canopy, it's spending 75% of its time absorbing all this light and not increasing photosynthesis. So it's wasted. And the paradox is that one leaf layer down, those leaves are light limited. And so the hypothesis of are plants too green comes from this notion that these top layers of leaves are absorbing way more light than they can use. But one leaf layer down, those leaves are light starved. If we could let more of that light in deeper into the canopy, it could be used with higher efficiency, and therefore we would expect more canopy photosynthesis that could drive a higher yield potential. And so this is the notion. And so here's our soybean plant. A full green soybean leaf has an absorptance for photosynthetically active light of about 0.9 OD. So it absorbs 90% of the light that's available. So 1 leaf layer down, it's 10% of the incident light. 2 leaf layers down, it's 50% of the incident light. And so if we look here then on our light response curve of photosynthesis, here we're way oversaturated. 1 leaf layer down, we're all down here in leaf limited. 2 leaf layers down, we're down here very close to the light compensation point. And so the very simple notion that drove the beginning of this program was, if we just do a simple calculation on Beer's law, if we reduce the chlorophyll concentration 70%, we still saturate photosynthesis here, and now the 2nd leaf layer down is close to saturation, and even the 3rd leaf layer down is contributing net photosynthesis. The spoiler is it's not this simple, as we'll see. And so this is the hypothesis: increasing light transmittance through the leaf will increase canopy leaf load. And so the way we wanted to start this out was to do a more sophisticated model and see what the model really predicts. And so we modeled this for a leaf area index of 4. And so that means having a canopy in which the area of the leaves is 4 times the area of the ground that it covers. And so soybean gets up to a leaf layer index of 7, 8, 9, wheat 6, 8, 9, something like that. And so this is modeled then for an entire day with this kind of profile, and it's modeled for a particular day a year, and it's modeled for 40 degrees north latitude, which affects the amount of solar energy that's available. And 40 degrees north latitude is essentially the latitude of the middle of the Corn Belt. And so when light comes in, it has 3 potential fates. It can be absorbed, shown here, it can be reflected, or it can be transmitted. And so, of course, what we're after is trying to increase the amount that's being transmitted to lower levels. And so if we calculate then for a full green leaf what the contribution is of each leaf to photosynthesis across the day, we get this kind of profile: leaf 1. Notice the change in scale. Now leaf 2, early in the day, it's actually negative to respiration. And then leaf 3, there's almost no net. In fact, it's all negative. And in leaf layer 4, it's all negative. And so then we can calculate what the net amount of CO2 that is fixed per land area. And this is what's really important when you're thinking about intensification. And in our model, then we can ask, so here's the full green leaf that's absorbing 80%, 86% of the incoming radiation. And what the model suggests is that if we reduce that to 55% by lowering the chlorophyll concentration, we should get about a 30% increase in the daily integral photosynthesis on a land area basis for canopy that has a leaf area index of 4. Okay? And so this is just looking at light absorption by a soybean canopy that we measured in Urbana. And so what you can see is that, and so day 190 is shortly after emergence. Then by day 197, we have a leaf area index of about 2.7. And you can see that there is, that the amount of light that's being absorbed is plateaued. We're absorbing about 95% of the total radiation that's available. And so there's little question that breeders have been able to optimize the canopy from the standpoint of total light interception. But our question is, has it optimized where that light's being absorbed in the canopy? And our hypothesis is that it hasn't. And so we set about, when we started this project, to look at a light green mutant of soybean. And there was 2 advantages of this. One, it existed and we didn't have to engineer it. And the second was that we were able to look at a plant that was isogenic for, there was isogenic other than this one mutation that existed in the chlorophyll biosynthetic pathway. We were able to show that the mutation here is in the enzyme of magnesium chelatase, which is an enzyme in the biosynthetic pathway. Clearly, it's not knocked down. Its activity is just knocked down. And so what we wanted to do here was to use the technique of light sheet microscopy to look at the penetration of light into a full green leaf versus the penetration of light into one of these light green leaves. And these light green leaves had a total chlorophyll content that was 65% lower than the wild type. And so they look like houseplants. And so they're really quite light green. And so what we do with this technique is from the top of the leaf or the bottom of the leaf, but the top of the leaf, we illuminate the leaf with different colors of laser light. And then from the edge of the leaf, we detect chlorophyll fluorescence. And so chlorophyll fluorescence, the intensity of the chlorophyll fluorescence is proportional to the amount of light that reaches that depth in the leaf. And we can do this for different colors of light. And so illuminating from the top of the light, if these are false colored, of course, but if we illuminate with blue light, you can see that almost all of the light is absorbed at the very top of the canopy. Red light penetrates a little bit further, and green light, we know that is reflected, and so it's not absorbed very well at all, penetrates quite deep into the leaf. If we compare that to the light green leaf that has 65% less chlorophyll, blue light's penetrating a bit more deeply, red light's penetrating a lot more deeply, and it doesn't affect green light very much. And so we can understand this pretty well. And so the two major chlorophyll pigments in higher plants are chlorophyll A and chlorophyll B. And so they absorb strongly in the red, they absorb very strongly in the blue, and they absorb very weakly in the green. And so we expect green light, because it's not being absorbed strongly, we expect it to be transmitted more, and we expect it to be reflected more. Blue light, which is absorbed most strongly, we expect it to be absorbed very strongly, and because it's being absorbed strongly, the amount of transmittance and the amount of reflectance is going to be low. And red light is intermediate. And so if we quantitate that, so we can quantitate those figures, and what you can see is what we saw qualitatively, that blue light is penetrating more deeply in these light green leaves, red light even more deeply, and green light not very much. These figures over here are illuminating from the bottom of the leaf, which we did because we could. And so then if we look at photosynthesis based on gas exchange as a function of the light intensity, we do see an improvement in efficiency of photosynthesis. But notice that this is APR, and so this is absorbed photosynthetically active radiation. So it's not incident. What I showed you previously was incident, and so this is what's being absorbed. And so what's happening, at least our interpretation, is that now there's less oversaturation in the chloroplasts at the top of the leaf with blue light and red light. They're penetrating deeper into the leaf to chloroplasts that aren't saturated, and therefore that light can be used somewhat more efficiently. Green light, it doesn't matter so much in a single leaf because it's already penetrating quite well, but also being reflected. And so I told you at the beginning, it's more complicated than this. And so the complication really becomes in this issue of reflectance. And so the difference between, and so the reason you get a lot of reflectance in a leaf is that there's a lot of air spaces in the leaf. And so when light comes in and it goes through a cell, it's going through an aqueous environment, that has a different refractive index than air. And so every time it goes into an aqueous space, comes out into an air space in the leaf, refractive index changes and there's light scattering. And it's doing this several times. And that scattered light, depending upon direction, can either be reflected light or it can be transmitted light. When you think about it, in our model of a leaf area index of 4, anything that you reflect from the top of the canopy doesn't have a chance to get reabsorbed by the leaves above it. And so it just goes back into space. And it's lost to the canopy as total solar radiation that's available to drive photosynthesis. And so that suggests that there should be a sweet spot. There should be a sweet spot of lowering chlorophyll to the point where you can increase transmittance, but not so much that you have lost total radiation, that you're actually lowering photosynthesis based on less light available to be absorbed. And so in our model, we can do a sensitivity analysis of, what are the most important things in determining, what what yield potential is going to be. And one of the things you find out is it's really this issue of the relationship of how changes in absorptivity reflect reflectivity and transmissivity. And so originally in the model, we were using this equation for dense canopies. And it's kind of an empirical fudged equation. And when we discovered how sensitive the model was to this issue of reflectance, we figured that we really needed to find out for soybean, what is the relationship between the optical properties of the leaf and the amount of chlorophyll that's in the leaf. And so at the University of Illinois, we have the USDA germplasm collection for soybean. And in that collection are 68 different light green mutants. A lot of them are light green mutants for really bad reasons, and they're really sick plants. But nevertheless, we have chlorophyll concentrations that go from 50 micromoles per meter squared up to full green of 500 micromoles per meter squared. And it so happens across those 68, they're pretty well spread out in chlorophyll concentration. And so we can go in then and we grew them all out in the field, and then we measured the optical properties, every one of those leaves, to get the relationship between chlorophyll content in a soybean leaf and how that partitioned into reflectance, how that partitioned into transmittance. And then we also measured a lot of photosynthetic properties from those leaves. And then we used that information in order to parameterize the model so that we were using actual measured values as a function of chlorophyll concentration rather than assumed ones. And what it shows qualitatively is what you expect, and that is a leaf transmittance and leaf reflectance increase with decreasing absorbance. And so here we see this relationship for leaf reflectance versus chlorophyll. And so chlorophyll concentration is going down in this direction, and this is wavelength. And so what you can see is that as chlorophyll concentration is going up, reflectance is going down, and vice versa in the red. And here you can see the green window. And then blue here is absorbed so strongly in both the full green leaf and the light green leaf that there's really very little reflectance predicted in the model. Here's leaf transmittance, and so this is what we're trying to maximize, and then here's total leaf absorbance. And so what the model predicts is that chlorophyll can be reduced up to 60% with very little seasonal penalty to crop photosynthesis. And so this looks at total light absorbed and total daily integral of photosynthesis as a function of day of year. And so this is as the crop's developing, and that's over what it is in the depth of the canopy. So anytime you see red, it means that the light green leaf is performing better. In the areas that you see blue, it means that the dark green leaf is performing better. And so this is then integrated here, and here's the full green leaf, and here's the light green leaf. And so it says that there is some penalty in terms of the total light that's absorbed over the growing season and any time during the growing season. And that penalty then in the model shows up as a penalty to net photosynthesis. And so it's interesting to know, and so this model suggests that plants, yes, have very much overinvested in chlorophyll, but it doesn't support a hypothesis that in fact we should see an increase in canopy photosynthesis. And so the model is not predicting that. And so this is modeled for the full green and this mutant soybean Y11. And so when we made those measurements in the field, we made them over 2 years. In year one of that experiment, if you look down here at seed yield, gram per meter squared, there's about a 10% penalty in yield. And that's, and so that's pretty, that pretty much validates what, what the model suggested. In the second year, this was 2012, and we had a huge drought. And so we saw bigger yield penalties in Y11 in year 2. Y11 does have a pleiotropic effect. And so even though the mutation is in magnesium chelatase, which is a 4 subunit enzyme that's involved in the biosynthesis of chlorophyll, It turns out that one of those subunits, which is in the envelope of the chloroplast membrane, is involved in ABA signaling. And this Y11 mutant isn't able to control its stomates, and so they stay open during drought, and their water use efficiency goes down. And so we're suspicious during this drought year that that's the reason. And so what we really wanted to know is if the model could tell us why. Why aren't, what is it that limits light green from not being a more productive canopy, but just a canopy in which we can get rid of a lot of chlorophyll? Getting rid of a lot of chlorophyll in and of itself makes some good sense, because if we assume that all of that chlorophyll A and chlorophyll B are associated with light harvesting pigment proteins of photosystem 1 and photosystem 2, and they almost certainly are, then we expect a 9% nitrogen savings for a 60% decrease in the chlorophyll concentration of the leaf. And so what the model told us was, well, a big issue here is the loss of photosynthetically active radiation from the canopy due to reflectance. And then there's, and related to that is this early season penalty. And so it's a time when there is a real disadvantage of being light green early in the season. And so two reasons for that. One is the canopy is small, and any reflected light has a lower probability of being intercepted. And then secondly, it doesn't do any good to transmit more light through the canopy if it's going to hit the ground. And then the third is that in spite of the fact that there is a nitrogen savings, it turns out that soybean aren't real bright. And so they don't take advantage of this extra nitrogen savings and reinvest it into photosynthetic apparatus in areas of the canopy where the light intensity is now higher. And so we have been successful, and I'm not showing the measurements, we have been successful of increasing light intensity lower in the canopy, but the plants aren't redeploying the photosynthetic apparatus to be able to deal with it. And so what can we do about that? And so this just shows this, and so I've showed you this before for the full green canopy, and now you can see for this light green canopy that there is this significant penalty early in the season, even though the leaf area index in the light green is actually a little bit higher than the leaf area index in the dark green. So there's much leaf area there, but there's higher reflectance here, and some of the transmittance is being lost to the ground. And so we wanted to be able to get around this early season penalty. One of the ways to potentially get around this early season penalty is not to turn on the light green trait until you have canopy closure. And so what we wanted then was to put an inhibition in chlorophyll synthesis behind an inducible promoter so that we could turn on the light green trait deeper into the season. And so the way that we started out to do that is to use an ethanol inducible promoter. Yes, we're out in the field spraying soybean plants with ethanol. Actually, this is being done in tobacco. And so again, we're expressing a small RNA, and that small RNA is targeted to magnesium chelatase. And this experiment is currently underway. And so this is a field experiment that's ongoing. Obviously, this is a drone photo of it. And we have, in each one of these blocks, we have 3 wild type, and then we have 3 each of two transformants with this ethanol inducible component. Both of these transformants work well in our greenhouse and chamber studies, but only one of the transformants responded to the ethanol spraying in the field. And we think it has to do with temperature, because we did do some greenhouse experiments where the temperature was high and this one transform didn't respond. And so we spray with ethanol every morning at dawn. And you can see after 5 days of spraying, we're now seeing in one of these transformants a consistent decrease in the amount of chlorophyll that's there. And so we're getting about a 40 to 55% reduction in chlorophyll following the ethanol spray in the transgenic plants. Here's this effect of the absorbance, the transmittance, and the reflectance. And so remember I told you that what the model predicts is we wanted to go from the wild green type of 86% interception of the light down to 55%. And so if we compare this, a 55% reduction in the amount of chlorophyll translates into a decrease in absorbance down to about 60% of the incoming light. And so we're very close to what the model predicts as being ideal. And this experiment is still ongoing. And so we do have green leaves at the bottom canopy. We are getting higher levels of light in the canopy. Whether this will translate into a canopy that has a higher biomass, we don't know right now. And so we're doing this experiment now, and we're planting a replicate experiment in Puerto Rico next week. And so we'll get 2 replicates of that this year. And so this is one of the ways that we're after to deal both with this issue of reflectance early in the season and this issue of lost absorbance early in the season. Lost absorbance both due to reflectance as well as transmittance through to the ground. And so the second thing we're thinking about of increasing the transparency of leaf, because that's what we want to do. We want to increase the amount of light that gets through it. Is there a way to increase the transparency without invoking as much loss due to reflectance? And so another potential way to go about increasing the transmittance of light is to take advantage of the Sieve effect. And so I'm teaching photosynthesis right now, and the way I explain the Sieve effect in class is that if you have an ink bottle full of ink and you set it in a bathtub full of water, you've changed the absorbance of that bathtub very, very little. If you take that same amount of ink and you divide it into 1,000 ink bottles, you've changed the absorbance more. And if you pour it in, then you've increased the absorbance most. And so that is a qualitative example of the Sieve effect. The Sieve effect is actually more complicated or more intricate, and that is that it has to do with the wavelength of light and the wavelength of lights that the chromophore is absorbing. And so in this example, if we take this to be a cuvette full of chlorophyll and we're illuminating with monochromatic red light, I've told you that a fully green leaf absorbs about 90% of that light. Which means, and so if you do that in the red, and remember I said, chlorophyll A and chlorophyll B absorb strongly in the red as well as in the blue Sorey. But if you do it in the green, then about 80% of the light is transmitted and absorbance is about 20%. Then the Sieve effect is to take all of this chromophore, take all of this chlorophyll, and put it in just half of the cuvette, and the other half of the cuvette just has solvent. Same thing down here. And now we have a transmittance here that's 0.5, but a transmittance here that 50%. And so now we're illuminating this whole cuvette with monochromatic light. And so this solvent is absorbing any of the red light. And this is absorbing more of the red light based on Beer's law. And now absorbance of red light for chlorophyll has gone from 90% down to 50%. But for green light, the effect on total absorbance is very small. The reason being it wasn't strongly absorbed in the first place. And so again, we're not interfering with the transmission of green light into a canopy. And incidentally, if you didn't know, green light drives approximately 50%, 55% of all the photosynthesis in a dense canopy, simply because it is transmitted into the canopy where it can be used more efficiently. Once you get to a leaf area index of 4, none of it's reaching the ground anyway. And so the notion that green light is inefficient in driving photosynthesis, may be true on a chloroplast basis, but it's certainly not true on a canopy basis. And so we wanted to try to take advantage of the Sieve effect. And so the way to do this in a chloroplast, if you go back to the ink bottle example, is to make fewer ink bottles, right? And so, rather than having 1,000 ink bottles, let's see if we can get down to 200 or 20 ink bottles. And the way we went about doing that is to interfere with chloroplast division. And so this has been worked out very well in Arabidopsis by Kathy Osterhold at Michigan State. And there's a number of proteins that are involved in chloroplast division, and one of them is FIT-C. And so we made an RNAi to FIT-C in order to lower its expression, and therefore, as she showed in Arabidopsis, you can pretty much titrate the size of the chloroplast based on the amount of FIT-C that you allow to form. And it appears that what happens is that total chloroplast volume doesn't change, total chlorophyll doesn't change, but the number of packets that you deploy it in gets smaller and smaller, gets fewer and fewer as the chloroplasts get bigger and bigger. And so this shows that some of her work. And I apologize to you for changing organisms, but this work was supported by DOE, and so we were working on sorghum and sugarcane in this case. And so Freddie Alpeter took our construct and transformed it into sugarcane. And so sugarcane is one of these C4 plants that has two kinds of chloroplasts. And here's the normal size of the chloroplast, and it has this volume. And we have lots of different transformants, and some of these transformants have chlorophyll concentration or plastic volumes that are 5 and 6 times larger. And so this should, based on modeling, have a significant effect on sieve effect and increase the transparency of the leaf. And based on modeling, it should have lower reflectance. And so this is the parental line. So this is, I'm switching again. This is actually sorghum in this case. And so this is the parental line of sorghum, doing Lightsheet microscopy as we've done before. You can see the penetration of the light into the leaf based on these different wavelengths. And so these are the laser wavelengths that have changed. And so when we increase the volume by a factor of 5, you can see that we are increasing the transmission of the penetration of these light deeper into the canopy. And so these, not these plants, but the sugarcane plants were planted out in an experiment that was done at Florida State by Freddie Alpeter. And we saw about a 10% increase in the biomass of these plants when they're planted out. And no change in the amount of sucrose accumulated. It's a little bit problematic because actually that increase in biomass is larger than we would predict in our models based on the amount of transmission. And so we're still trying to work that out. So just to summarize, what I told you today is that field experiments with soybean and with tobacco have validated our canopy model that there's almost no penalty in light absorption or carbon gain with a 60% reduction in chlorophyll. And that 60% reduction in chlorophyll is equivalent to a 9% savings in nitrogen. It's interesting, in all of these plants that we've measured where we've reduced chlorophyll, even though we've been able to show that they're not investing in chlorophyll, they're not investing in those chlorophyll proteins, those are gone, the C to N ratio hasn't changed. And so they're partitioning this extra nitrogen into vegetative storage proteins, at least in the case of soybean and tobacco. And so the nitrogen is still there. These are grown under very nitrogen replete conditions, but they're not reinvesting that nitrogen in ways that would help the plant. And then the canopy identified 3 major limitations that prevent the light green trait from actually improving canopy photosynthesis, improving yield. As I said, they were lost due to reflectance, this penalty of light green, and that the light green plants don't reinvest their nitrogen in increasing photosynthetic capacity in those areas of the canopy where the light intensity is going up. And so what are the next steps? And so I've told you one of them, and that is to use light-inducible promoters to try to sustain the full green phenotype until canopy is closed and then induce the low green phenotype. Obviously, our plan is not to do this long-term with ethanol sprays. Postdoc doing is pretty tired of getting up at dawn to spray. And so we either want to use a phytochrome promoter or there's a group that we're collaborating with Germany who believe they have a promoter that actually responds to light intensity. And so either of these potentially could work to get the phenotype that we want, which is light green at the top of the canopy, dark green at the bottom of the canopy. The other thing we want to do is that our models suggest that combining low chlorophyll with a large chloroplast trait should be the best of both worlds in terms of increasing transmittance and minimizing the increase in reflectance. And then finally, it's this issue of nitrogen nutrition. Now, I just mentioned that all of these experiments have been done under nitrogen replete conditions. So in the case of tobacco loves nitrogen. And so it's actually grown at twice the amount of nitrogen that we put on maize. And so maybe it's the case that under these luxurious nitrogen conditions, the plant doesn't have to make a choice and therefore is, just keeps its same nitrogen investment. As I said, well, maybe I haven't said, but much of this work is supported by the Gates Foundation, and their interest is really in not high input agriculture, but their interest is in the agriculture of sustainable farmers that are growing in these very bad tropical soils, and they don't have access to fertilizer. And so our hypothesis that we want to test is that under conditions when nitrogen is very limiting, that plants may take advantage of this nitrogen savings and reinvest it in a way that actually takes advantage of this change in the light intensity. That isn't that they're going to do better than the high nitrogen situation, but compared to a full green, low nitrogen plant, the hypothesis is that they will do better. And so with that, I'll stop and entertain questions.
I was wondering if using your models, whether you can see in the canopy where you have reduced chlorophyll at the top, if you can see whether you're interfering with the partitioning efficiency, especially into yield or into the seed.
Yeah, and so the model is assuming partitioning efficiency, and so that's not a variable in the model. We could put it as a variable model if we could experimentally parameterize what that would be. And so what the only field experiments that we've done with light green that I'm confident in the harvest index is with the Y11 light green soybean. And in that case, we do not see a statistically significant change in harvest index. Whether that's going to be true over a wide range of light green plants, and particularly when we make that cross from leguminous plants to non-leguminous plants, we don't know.
In the beginning of your talk, you showed us a slide and it gave us the perspective of a horizontally positioned leaf at right angle with the incoming radiation. And then you did some modeling on how lowering chlorophyll might influence penetration. Could you model what the effect would be of having angled leaves that are more vertical positioned, which would be another way of getting exposing that leaf to a lower flux density that's not saturating and allowing more light to penetrate?
Yeah, and absolutely, you're completely right. And so another way to get at creating better homogeneity and light penetration in the canopy is to change leaf angle. And so there is work going on in our Gates funded project to look at both natural variation in leaf angle as well as engineered variation. And so in maize, a number of the genes that are involved in leaf angle are known. And so in fact, that's not in the right project. It's in a DOE project for the C4s. And so both natural variation in that is being looked at as well as genetic variation. And then, the plan is to, if we can work out this light green trait, is to try to partner the two. Even in soybean, there is a lot of genetic variation in leaf angle and leaf shape. And Lisa Ainsworth is doing a natural variation looking across a big diversity panel in soybean to look at that. And, you know, and what she finds early in the season is that it does matter. And once you get to a big leaf area index, it doesn't matter. But early in the season, it
And
does matter.
soybean has this ability to para-heliotropism.
Yeah, and so a lot of that has been bred out in the most modern cultivars, but yeah, in some of the historic cultivars, it's really a problem. And it becomes a real problem to model too, because in order to parameterize the model, you have to know what the leaf angle is.
Change the nitrogen remobilization enzymes, like play with those, like a stay green phenotype, a stay green phenotype, or grain protein content, like mix the pale phenotype with those.
Yeah, and so the question is, what about a stay green phenotype? And so a phenotype that stays green longer into the season and increases essentially the growing season and the period of seed fill, depending upon, you know, what crop it is. And if it's the kind of stay green in which the plant stays green and stays photosynthetically active, in fact, that has been shown to increase yield. And the reason it increases yield, it increases S, you know, in that equation, it increases the total amount of solar energy that's collected during the season. And so, you know, have we thought about combining that? We have, and the genes are known to do that, but we've not started that one.
So as you're reducing the chlorophyll in the top piece, I'd assume you're also exposing the leaf to more potential for photo damage because it can't quench the excess light, especially in the upper canopy level of the light. So if you looked at how photo damage chlorophyll content.
Yeah, and so the question is about, is there a change in photo inhibition or photo damage that is a function of absorptivity? And so our hypothesis was actually that the amount of photo damage would go down because the amount of total light that's being absorbed. And so one of the main ways in which plants deal with all this excess light that I showed you that they get during much of the day is through a process that's been unfortunately dubbed non-photochemical quenching. And so it's a process by which excited chlorophylls can be de-excited through some carotenoids and released as heat. And it's quite easy to measure because you can measure it by a fluorescence technique. And so we expected the amount of this non-photochemical quenching to go down, and it didn't change. And so again, this is something that is contrary to what we think the plant should do, but it didn't care. But that's also engineerable. And so, you know, you can engineer the amount of non-photochemical quenching that's done because the genes that are involved in doing that are known, and it's already been shown in model systems that you can manipulate it. And there's also natural occurring genetic variation.
Don, you know, in maize, there's been, 50 years worth of selection where most of the selection was on plot level performance.
Mm-hmm.
And so, they didn't necessarily know what they were selecting for, but they were selecting on that plot. Is there any evidence the transmissibility of light in elite maize of today is greater than it was 50 years ago?
And I would say no. If anything, I would say it's the other direction, that things have gotten greener. And so if you'd asked me this a year ago, I would have put the blame on you breeders and saying that you guys don't like things that look like green, and so you rip them out. And then the second thing is, depending how big your plot is, it could be shaded by a full green. But what I would say now is that our modeling predicts that it shouldn't be any different, right? And so what I think is that I would predict that you would see a very small penalty for being light green in those trials, but not something that you would pick out as something you wanted to carry forward. And so if it is going to be successful, it's going to have to be something that that stacks a couple of different traits to try to circumvent these things that we think are, preventing the light green trait from being more beneficial.
there, you know, if you go back over that 50 years, those first couple of generations of seeds, you know, before we made directly DH and direct GS, it wasn't exactly top level selection, but I think there is still an opportunity there for that individual shading competition to play a role. So I'm kind of wondering, actually, now the last 20 years with all DH, all GS,
You probably don't have that variation.
the individual's performance no longer matters. Yeah. And it actually things that we haven't seen yet really in our hands. Yeah. May have changed.
I mean, I guess in reflecting the question was asked over here about leaf angle, my impression is that in maize, leaf angle has gotten closer, but at the same time, crowding tolerance has gotten better. And so maize breeders have taken advantage of that, and so light penetration hasn't improved, but density's improved. Is that fair?
Yeah, I think that, but I would also imagine they actually, like all these strategies, you know, selection may favor, and I'm just kind of wondering which density, those first few generations of making an inbred line, you wouldn't be planning at today's density for when you're selfing down the seed over the last decades.
Susan?
Have you tried, I mean, maybe not yet, but have you tried looking at what, let's say, low nitrogen or low input agriculture is doing as opposed to what high input agriculture is doing in terms of selection? Because when you have those other constraints, it may totally rewire the.
And we haven't, and so, you know, as I said, we're planning to do that in the tobacco experiment, but maybe we should do it in the mutants that we know much more about. But the mutants that we know a lot about are leguminous. So.
But I'm thinking about the fact that in rice we have varieties, they're traditional varieties that are grown under low input, and their yields are nothing like what they are under high input. But the selection has been over many, many, many decades, right, for performance. And I'm just curious if once you set in place other limitations.
Yeah, and we actually could do a lot of that in our canopy model. And so the canopy model is driven, you know, by meteorology that we measure, but it's also driven by parameterization of what we put in terms of, you know, nitrogen supply, as well as all the measured parameterizations of photosynthesis. And so we could put that in and take some tweaking in the model, but that's a good suggestion. Yeah.
Does it make any sense in a sort of general way to work in the opposite direction, at least with some crop plants, and increase the light reception in the top leaf and decrease the number of leaves so that you get a gain in carbon?
And so if we run this same canopy model, which we can do, and ask the question, when parameterized for full green soybean, you know, under the growing, you know, parameterizing with all the meteorological data from a growing season in Illinois, it turns out that the ideal leaf area index for a soybean plant is 4.1, and so it often goes to 8. And so you're right. The model suggests that it very much overinvests in the amount of leaves that it's producing. And you know, why you think that might have to do with respiratory load, the model suggests that the real reason is investment in making that leaf when you first grow it. And so Steve Long's group did an experiment where they went out and they clipped every third trifoliate from soybean plants before it was allowed to expand. And again, soybean plants are not very cooperative, and so as soon as they did that, they branched more, and so they weren't getting the depression and leaf area index that they wanted. But they got it from 8 down to 6. And when they got it from 8 down to 6, there was a statistically significant increase in field experiments, not field trials, of about 10%. And so, you know, that's not defining, you know, and again, I don't think farmers are going to send out 25 undergraduates to clip their soybean fields, you know, but maybe there are some genetic controls that can do that. And so absolutely, at the planting densities that we're using, I think most crops are over-investing in leaves after canopy closure.
One more question.
can you achieve the same goal by careful management of nitrogen? Let's say you develop a nitrogen management regime that allows very rapid canopy diphtheria development early in the season. And then once the canopy is fully developed, you lower the nitrogen. So the leaves will become light green. Can you do that?
Well, I suppose you could, but I would guess that there would be a yield penalty associated with the nitrogen that's available to put in to the seed during seed fill. I would think in something like rice or in wheat, that would be disastrous, right?
Soybean.
Well, soybean, it's a difficult experiment because normally in Illinois where we grow soybean and maize on an annual rotation, there's enough carryover of nitrogen from the previous soy crop that they don't nodulate and begin to fix nitrogen until late July. And so I think if you did that experiment in soybean, they just nodulate way earlier. There are non-nodulating soybean though, so that would be possible.
Good, thank you very much.
This has been a production of Cornell University, on the web at cornell.edu.